imagine a world where we have developed novel therapeutic approaches for some of the most deadliest diseases present. Imagine a world where we no longer have to worry about the genetic disorders that come our way. The things that you have imagined just might soon become a reality, and all this would be possible with the ever-booming technology of gene editing. Gene editing is a very specialized technology that involves cutting, pasting, and altering our genome. To be more precise, it involves altering our DNA at certain sites. Being a very specialized technology, the tools used for this technology are also highly specialized. Today, in our Interview with Scientists series, we have Dr. Saikrishnan Kayarat, the 2019 Shanti Swarup Bhatnagar award-winning scientist from IISER Pune. He works on one of the highly specialized tools of gene editing. Today, he is here to share his research and experience with us. Welcome, Dr. Kayarat, and heartiest congratulations for winning the Shanti Swarup Bhatnagar Award.
Thank you, Gargi.
So, talking about the gene editing technology and the tools that are used, so can you please acquaint us with what tools do you and your team use for this technology?
I'm a structural biologist and I primarily use tools of structural biology, that is X-ray crystallography and cryo-electron microscopy for the studies that I do. In addition to this, I also perform biochemical and biophysical assays to understand the system that we are working on. So, tools that are used for biochemical and biophysical assays are used in my laboratory extensively.
So, obtaining your PhD from Indian Institute of Science, Bangalore, and then a postdoc from Clare Hall Laboratories, UK, how has your research changed from being a PhD student to being a faculty at IISER?
The research goals have remained the same. The questions have been the same. The question has been to understand how proteins work. And that is a theme that has continued since my PhD days. Before PhD, I did a bachelor's in physics, and then I got this interest in science, biosciences, and I joined for my master's in Indian Institute of Science through an integrated PhD program in the life sciences. And that's where I got this bug for understanding how proteins work. And that has been the main theme since then. How my research activities have changed, that has changed enormously. As a PhD student, you are focused towards understanding the question, which is the research question that you have to address for your thesis. You are very focused, and that's what you're interested in. In addition, you are interested in you're involved in extracurricular activities and also in understanding science in a broader sense. As a faculty member at IISER, we are involved not just in carrying out science, which involves not only addressing questions and also being involved with the research students to understand and take the questions to their logical conclusion, but also we are involved in many other activities of the institute, the responsibilities that are there in developing the curriculum, in developing the institute per se. So, we are involved in many other activities in addition to carrying out my research work. And one of the most important activities is teaching, which I was not involved as a student, but now I am deeply involved in.
During your PhDs, on which topic were you working? And as many scientists or many researchers change their field from PhD to coming into academic field, but your topic has been, I guess, same. What motivated you to keep on going with the same topic?
So, as you mentioned, my research theme has focused on understanding how protein structures, what protein structures are and how they work. But the model systems that we have been studying, the systems that we have been studying have changed over the time. During my PhD time, my research focused on proteins from mycobacteria, and our laboratory was one of the pioneers in establishing the structural genomics program of mycobacterial proteins, which was an international consortium, and our laboratory then, the PhD laboratory where I did my PhD, was involved in establishing this consortium. And I was involved in determining structures of proteins, a few of them, which were involved in replication and translation. I was involved in determining the structures, and that formed the basis of my PhD work. For my postdoctoral work, I moved to Cancer Research UK, as you mentioned, Over there, I worked on DNA repair enzymes and how they function. The tools that I use, that is protein crystallography, remained the same, but the systems then changed. And the DNA repair enzymes, how they work, was the research project that I undertook, and that's what led to my publications during the postdoctoral work. From there, I moved to IISER Pune, and at IISER Pune, I established a program which is different from what I had done for my postdoctoral or PhD work. But interestingly, this was influenced by my master's project, and that's the project on the restriction modification enzymes that we have established in IISER to understand how they work. And towards that, we use protein crystallography and cryo-electron microscopy, in addition to all the other tools that I mentioned. So the theme has remained the same, but the questions that we are addressing has changed.
Okay, so which is also necessary in research.
Yeah, what drives research is the question that you are addressing at any given time point. And what drives those questions or how you can tackle those questions depends on your expertise that you have gained over the course of your research career.
Okay, so and your latest accomplishment, the Shanti Swarup Bhatnagar Award. So how rewarding is this for you?
Well, it's an important award for an Indian scientist, and the fact that what I understand is that the award is given after a peer review of the work, which is by scientists all over the world, a team of scientists from all over the world. Their reviews are considered, and then a panel of scientists from India who then select the awardees. So what it means is that our science has been recognized, the quality of science that we do has been recognized. What it also is important is that the hard work that my students, the PhD students of my team, past and present, the BSMS students who have carried out the thesis projects or semester projects, the hard work that they have put in has been recognized and been rewarded. So I think it's an important reward that we have got.
Okay, so again, do you think collaborative work yields more fruitful results than individual work, or?
It depends on the field of research. It depends on the area of research. It depends on the question that is being addressed. It depends on many other aspects to address this question whether collaborative research is important or not. In a field of research like mine, where I try to address a question using a certain technique, collaborative research can be very helpful if it can provide complementary information that the techniques that I employ or the expertise that I employ cannot on its own solve it. So complementary information that can be derived and answers the question in a better way is surely going to benefit science. After all, what we are trying to do is address questions of science. If collaborative work can address those questions in a better way, I think collaboration is an important aspect. And science cannot wait for an answer.
Yeah, that's true. Okay. So what are the analytical techniques that you use in your laboratory?
As I mentioned, the tools that I use are X-ray crystallography and cryo-electron microscopy. These are the tools that we use for determining structures. Towards determining structures using X-ray crystallography, one has to grow protein crystals or complexes of protein with DNA, which is what we do, and that is an aspect that we extensively work on. We have to study the biochemistry of the proteins to be able to crystallize this protein molecules. So my students and I are spending a lot of our time understanding the biochemical and biophysical properties of this protein. So tools like protein purifications, techniques like protein purification, their analysis, say simple analysis by SDS page or something, and to high-end like single molecule techniques are techniques that we use or collaborate with to use.
Do you think there are limited resources for carrying out the biological crystallography in India?
Indian science has developed quite well, in particular biological crystallography, macromolecular crystallography, and the development has resulted in us having infrastructure to carry out crystallographic studies in-house, that is within an institute or within a laboratory, through diffractometers that are available in many laboratories, including IISER Pune. And that has made it quite a technique that is accessible to many users. Having said that, something that Indian science had been missing for a very long time, in particular biological crystallography, has been a synchrotron source for macromolecular crystallography. We have a synchrotron source now in indoor, which is an older generation, which I think is a great achievement that we have built such a synchrotron facility, and which is serving the community. But what we are missing is the next generation synchrotrons, which are there all over the world. And fortunately for us, the government of India, through funding bodies like DBT and DST, have allowed access to such facilities abroad. So during my PhD time, when I did protein crystallography, I solved these structures. I solved the structures using diffractometers, which were in-house. I didn't have access to synchrotrons abroad. If I had then access, it would have shortened the time of determining the structure or possibly improved the resolution of the structures. But now we can do that because we have access to such facilities through these programs. And of course, what involves is more work than what our colleagues abroad would be doing. We need to grow crystals, test them in-house, take them, ship them abroad, which is not a trivial task considering the immigration, customs and other clearances. Not the immigration, but the custom clearances. It is not an easy task. Immigration also sometimes if the scientist is traveling along with the crystals. So it is a lot of hard work and logistics involved. And that is something which we may be able to tackle once we have a synchrotron of the next generation in India.
So, how will you brief about your research in the language of layperson?
My laboratory is interested in understanding how restriction modification enzymes work. Now, restriction modification enzymes are bacterial defense mechanism. They are bacterial defense against bacteriophages that attack bacteria. Just like human body is attacked by viruses, interestingly bacteria have their own viruses that infect them and kill them. Now, to prevent the viral attack, just like we have human immunity, there are systems which are now often dubbed as bacterial immunity. This immune molecules prevent the bacteriophage from entering or destroying the bacterial cell. What restriction modification enzyme does is it cuts invading foreign DNA. Invading foreign DNA could be bacteriophage DNA or any foreign DNA that can enter the bacterial cell. And they work as molecular scissors. They cut the DNA into pieces. Once the DNA piece is being cut into pieces, the genetic information of the virus is lost and it cannot grow within the bacteria cell and nor cause infection. Now, what we are trying to do is understand how this restriction modification enzymes work. And the restriction modification enzymes were discovered quite way back in the 1950s and 60s. And actually, it has led to 3 Nobel Prizes over the years and has been the foundation for understanding how proteins interact with DNA. So having said that, the systems that we study are quite different from the systems that have been studied so far, in the sense that the molecular scissors that have been studied so far are just scissors. They cut the DNA at a specific point. The scissors that we study are actually analogous to a machine, where the scissor is driven by a motor, and the motor requires energy, and the energy is nothing but the chemical fuel that is within the cell. So the scissor is driven by a motor, and how this motor driven scissors work is what we have been trying to understand. As I mentioned, the kind of studies that we have been carrying out has not been happening for such a long time, primarily because the enzyme systems that we study are quite huge, and it's quite difficult to study them either structurally or by physically. What my students and I have developed are ingenious techniques that can help us purify large quantities of this protein to carry out structural studies on them as an intact complex. And that is what we do in the laboratory to understand how these machines work. Now, why should we one determine the structure to understand their function? The reason that one should determine the structure is because the structure of a protein determines how it works. A protein is essentially nothing but an arrangement, a three-dimensional arrangement of atoms. And this three-dimensional arrangement of atoms is what allows it to carry out the function that it does within a cell. And it is important to understand what is the three-dimensional arrangement of this protein molecule to understand how they work. It's analogous to, I told you the proteins that we work are similar to molecular machines. And to understand how a machine works, one requires to have a blueprint. And what we determine is essentially the blueprint of the machine in atomic detail. And that's what we do in the laboratory.
So, and what are the future prospects of your research, or how it will directly benefit our society?
If you look at science, the findings of science can be of two kinds, basic and applied. Humankind has always been driven by a curiosity, driven by a need to understand what is life, what is nature, what is universe, and many such questions. We are contributing to that in a small way, how something in a bacterial cell prevents viral attack from happening. That's the basic question that we are asking. We are also asking how complicated protein molecules work. That's another question that we are asking. And this is a contribution to our intellectual curiosity. Having said that, there can be applications, and we are acutely aware of that. And a part of my research team is involved in trying to see the technological benefits that can be derived from such molecules. And as you said, the molecules that we are talking about can modulate DNA. They may cut the DNA, they can chemically modify the DNA. That's what all these proteins that we work on can do, which in essence means you can modulate the genomic information, the genetic information within the genome of an organism. And we are developing techniques or trying to see if we can use the protein molecules that we study, if they can be modified to carry out modification of the genetic information. This need not necessarily be by cutting the DNA. This could also be by modulating what is called the epigenetic landscape of the genome.
So, what do you look in a student when selecting a PhD student? So,
it's a hard task to decide who is a good student or who is not a student, or a student who may not be suitable for the project that one is undertaking. It's not a trivial task, but we have to make that decision in a short period. And often I go by my hunch if the student is passionate or at least sincere about the questions we are asking. So sincerity can be gauged if the student has understood the questions that we are asking. It becomes very clear during discussions with the student whether they understand what we are doing. And sincerity and passion is what drives the student's research project. And if there is a hint that the student is sincere and dedicated, I think it's a good enough sign to take the student for the project.
what career advice would you give to students or young minds?
Again, it's not something that is it's not an easy thing to do to advise. Every student, every person is different from the other. But what is true is that success in whatever career that you choose would come if one is passionate about it. There are two things about a job. It could be a vocation or it could be an occupation. Now, if your job is a vocation, which is essentially what your hobby is also, what you're passionate about also, you would be really successful, because you are no longer constrained by the time decided by some authority for carrying out the job. You do the job whenever you feel like, and you're almost always at the job. So I think go for what is your gut feeling, what your passion is all about.
So, where do you think India stands today in science communication, and how can scientists contribute to science communication?
I think science communication in India is developing tremendously. Like this facility at IISER, it's pioneering science communication. What we have now achieved is outreach of science and communicating science to the students, and that is going on very well. What should be the next step from my perspective is communicating science to the person in the society, a non-scientist in the society, not just students. And that is important because it is important for a person in the society to know what is the significance of science, why should science be funded, and also to understand what could be the long-term benefits of science. And that requires a proper outreach by the scientists themselves, which requires that the scientists convey their ideas of their research or the results of their research in a layman's term and excite them to think what its benefits could be for the society. And that's something, in a way, scientists can contribute through the help of media centers, through the help of media itself, and so on and so forth.
So, as a kid, who was your inspiration during your school days or college days, and who inspired you to take up science?
My parents were, my family was, or when I was growing up, my family was not into science. They were, science was not something which was spoken about, talked about in the family. But then the first impression on, or my passion for science started from, it may sound mundane, but from CBSE textbooks. And in the CBSE textbooks then, I hope it's still there used to be nice colored boxes, where the boxes would carry stories of scientists who made a discovery. And it was a story that really inspired me. Unfortunately for me, my brother and my sister both did their bachelor's in science, and I was the youngest in the family, and I got to see the books, and the books really inspired me the next level. And my brother then went on to do a PhD, and it was clear to me that that's what I also want to do. And my brother then went on to establish in his lab in University of California, and I knew that I also wanted to do that, but I thought I will do it in India, and that's what I did. So this has been what my trajectory has been in terms of the inspiration at the as a kid. So it has been influenced by my textbooks, of course the teachers who encouraged us to read these books and also pointed out to this, and my brother.
Thank you, Dr. Kayarat, for your valuable time and sharing your research and experience with us.
Thanks, Gargi.